Then I think we can then start. And thank you. Good morning, everyone. My name is Roberta Piratelli, and we will be the moderator of today's webinar. This webinar is one of the activities of Instruct-ERIC that, as you know, is a pan-European distributed research infrastructure making high-end technologies and methods for structural biology available to users. The webinar of today is dedicated to presenting some of the innovation that have taken place at our center in Italy, the Instruct Center dedicated to NMR spectroscopy. And this improvement, enhancement, and also diversification of our services has been made possible thanks to some national funding that we received from the Italian Ministry of Research within the Ithaca SB project that has been dedicated to empowering our research infrastructure. And of course, this support has enabled us to strengthen our technological capabilities and also to broaden our research activities and also the services for the users. We will also have the opportunity today to present some of the results of some JRA activities that we have carried out lately within the Isidore project. So we will have 4 contributions, one more interesting of the other. So I will start immediately with the one by Lucia Banci, and she will present about the broadening of the NMR landscape for structural biology at Instruct-IT. So please, Lucia.
Yeah, thanks a lot, and thanks for the occasion to present, as Roberta said, the development, the improvement, and increase and expansion of the Instruct Center in Italy. And the Instruct Center in Italy is based on CIRMMP in Florence. And you can see from this picture from the sky that is featuring a number of quite a large number of NMR spectrometers. So the flagship technology is NMR, both solution and solid state, and as well as a number of other techniques starting from molecular and cellular biology, different type of biophysical techniques, other spectroscopy like EPR and so. So this is the core is NMR, but is flanked by many other techniques. And these are the type of services that are being provided by the Italian Center of Instruct, and where the flagship, as I said, is mainly solution NMR, as well as a solid state NMR. And specifically, we have received with the expansion and the funding from the Italian government, the very fast spinning probe for the 1.2 gigahertz NMR instrument that is the highest field in the world. As Roberta mentioned, this is the project Ithaca SB, which was set up to expand, reinforce the Italian the services in structural biology to the Italian community. And so there were support and expansion of the Florence Center, the Italian Center of Instruct, as well as other centers in CNR in Italy, which expanded various other techniques complementary to NMR and other structural biology techniques. Specifically in Florence, we have expanded the service in solid state and in solution NMR, in protein production and sample production in general, biophysical characterization, as well as was performed a series of training events. Going to the specific type of services which were extended in the due to this major contribution regarding solution NMR, it were implemented a effective, very high, very powerful and useful fluorine NMR probes and phosphorus 31 probes. So then we were set up a new spectrometer, NMR spectrometer, which was equipped for solution experiments with the quadruple cryo probe, which has 4 coils. In other words, we can excite and monitor 4 nuclei simultaneously in the experiments, so we can detect proton, nitrogen, carbon, and fluorine. And in a similar probe with this for phosphorus installed at 500, and so this is particularly useful for characterizing nucleic acid by themselves, interaction with protein ligands, and so on. And all these experiments can be done in vitro, but also in cell, and Enrico will present the application and what we can really gain with this type of application. In addition, we have also another probe, is a 3-channel probe where a coil can alternate, detect either proton or fluorine together with nitrogen and carbon at 700. So different magnetic fields and different type of probes, which can really expand the range of field nuclei which can be detected. Another important issue we have implemented is the equipment for measuring this diffusion order spectroscopy. This is a technique based on the application of gradients. I don't go in the details of the technique, just only what we can gain. So we apply gradients to the sample, and then this allows to monitor the diffusion rates of molecules in solution. And so this allows to characterize in a complex mixture the molecules that they have different size, as well as, for example, the formation of complexes. And so then if the protein is isolated, when you mix with another protein, if it's or other protein or biomolecule in general, if it forms a complex. It's important to stress that it can be applied also on unlabeled samples. So these are an example of a combination of diffusion experiments together with heteronuclear experiments. So in this case, you need label samples, and in this specific case, being characterizing the different diffusion rates of these specific proteins, for example, in different experimental conditions, and particularly in this protein, it undergo equilibria between folded and unfolded, and so then the signals for the 2 sets of conformations are characterized, and then the different diffusion rate of the 2 forms can be characterized, in particular in different experimental conditions. But as I said, it can be also used for characterizing the formation of complex and the specific protein. When is made to interact with the partner protein, you see a change, a variation in the diffusion rate, and then this indicates the formation of the complex, and then you can characterize the species, and to get information of the hydrodynamic properties. And as I said, it can be performed also on unlabeled samples. Other aspects that we have extended, including NMR, but also protein production and sample production and cellular sample production at different level, is also for the inclusion of specific labeling in the amino acid of different types, and this is particularly useful for in-cell NMR experiments, and then Enrico will talk about that. And also the use of a high very high, ultra high magnetic field, as well as the new tools and the new console, which allow very peculiar experiments, or very effective in terms of efficiency. It can allow to characterize, to perform a high order structure, so to characterize the biologics and their properties, even without, again, labeling. And Marco Fragai will present application of this. If we go on solid state NMR, as I said, we acquired the 0.7 probe for this for solid state, and this allows to characterize the very high resolution, also proton NMR spectra of solid state sample. And this was installed on the 1.2 gigahertz, which is operating in Florence, open to services since 2020. We were the first to get it, and this already since several years that is available to users. Now is open also the service for solid state. And as I said, the advantages of having this setup is that we combine the improvement that we have with high magnetic field and with fast magic angle spinning, because with this 0.7 is the size of the rotor of the sample holder, and you can see that the size is incredibly small, but being this way can spin around this so-called magic angle very, very fast, and average a number of couplings, which usually make the lines in the solid state spectra too broad, and increase resolution. So having this ability to spin so fast, we increase the sensitivity, we increase the resolution because we reduce the line width, and having the ultra high magnetic field as well, we increase sensitivity because we have a higher magnetic field, and we increase the resolution because we have the spreading of the signal over a larger range of frequencies. And so this allows to characterize with high resolution also proton. These are carbon spectra, and okay, this specific example shows how the type of information we can get for solid state for molecules which are not soluble, which are too large for the solution, but we can get a very high resolution characterization of the, for example, in this specific case, the formation of the complex and the regions which are interacting. And in this specific case, that these are spectra acquired with a new probe very recently installed, that it allows, you can see that on the proton dimension, you can really monitor the individual signals. You don't have overlap, and this is one human protein, and there is the wild type and the mutant, and they can clearly detect the shift and the changes in the spectra, mutant and wild type, and characterize the region which are involved in the aggregation of this specific of the protein. The mutants induce tend to induce protein aggregation and form and undergo amyloidosis, and so then you can clearly monitor the way of interactions of these various species. And so then you should appreciate for NMR people the high level quality of the spectra that are collected in solid state and not in solution. They approach the quality of solution. Other type of services that overall CIRMMP are providing is services for metabolomics, and has been, for example, extensively involved and active within also the Isidore project, and as well as beyond this. And about this, Paola will present example on the relevance and the power of metabolomics to characterize biological processes. Other techniques as well, relaxometry, and relaxometry is an instrument which measure the water relaxation properties, the nuclear spin water relaxation properties, has a function of magnetic field going down to very low magnetic field. And this provides information on the hydration of the molecules as well as the size of the system. And so they very nicely integrate with the dosing experiment. And so this can provide a really detailed characterization on the conformation properties, the dynamic features, as well as the hydration properties. And this also is being upgraded to a new instrument. Further, in terms of spectroscopy, EPR, you know that Instruct-IT is already providing services to EPR, electron paramagnetic resonance, which is detecting the transition between electron spin levels. So then is applied to paramagnetic molecules as well, molecules which were modified the spin level. And using also pulsed experiment, we can measure long distances between two different electron spins, which can be put as tag on large molecules. And these are measurements done at low temperature, so it is independent of the tumbling, can be done like in solid state, that it can be also slow moving rotating system. What has been expanded is this Ender type module, and this Ender measure the electron nuclear double resonance. So it measures the coupling between nuclear spin and electron spins. And so it measure the coupling and can derive the distances between these spins. And in particular can detect nuclei which are strongly coupled with the paramagnetic center, because weakly coupled we can measure through a classical paramagnetic NMR, but those that are close around the metal center, the paramagnetic center, like the ligand themselves, are in number of cases hardly detectable from paramagnetic NMR. Not all in a set of system we can, but in other not. And in this specific case, with a copper bound, this would be undetectable from the NMR point of view, but they can be detected from this type of spectra. This is electron spin resonance, and this is a Ender spectrum, where you see this is radio frequency. So this is the frequency of nuclei, and where they are subjected, these spectra are acquired when the sample is subjected to a microwave, that is the frequency of transition of electron spin, and this is then are excited the nuclear transitions. And here we see the coupling. Now it's require analysis, but just to give a flavor, here these are the weakly coupled protons to the paramagnetic center, so they have to acquire all that are relatively far away. And here we detect the coupling with the protons very close to the paramagnetic center. And here we see also the water molecule, that here are frozen and block the water molecules. So these are the protons of the water molecules that are bound to the metal in the protein. And for example, in the specific case, adding an oxalate anion that is known to remove the water molecules, we see that disappear the signals and the reduction of the coupling. And so then we can, from the analysis of the spectra, understand and determine the electron spin delocalization and characterize also the electronic structure that is a vital aspect in understanding and describing the functionalities, the properties of the metal ion itself. Has been improved the biophysical, expanded the biophysical tools for the characterization of biological systems, as well as the protein production, and Marco will talk about expression of viral proteases, proteins, specifically for drug development, and the mammalian expression for in cell NMR. And with this, I thank you for your attention, and I am happy to take any question. Thank you.
Thank you, Lucia, for this very nice and complete overview of the services that we can provide and the new installations that we have done. I don't see questions in the Q&A window, so I don't know if you're...
At the end, probably...
Anyway, yes, at the end we will have anyway some time to answer questions also on the general aspects that Lucia presented. So in the sake of time, I would go ahead with the presentation by Enrico Lukinat, who will present about time-resolved NMR to investigate drug binding and metabolic changes in living human cells. Prego, Enrico, try to share your screen.
I'm sharing my screen. You should see the presentation.
Thank you.
Perfect. Okay, thank you, Roberta. So I will also provide a brief overview of the recent technical development that we will make available as part of the Instruct-ERIC platform for in-cell NMR characterization. So, and then I will focus on an application, an example of an application to protein-ligand interactions. So we've been doing this for several years now, and in-cell NMR is a great tool to provide information within the cellular context. So we've been focused on the human cells specifically, so a lot of protein targets which are interesting. They are intracellular proteins expressed in our organism, in our tissues. And the way this is done in CIRMMP in Florence is by overexpressing the protein directly in the human cells. So for NMR characterization, as you all know probably, we need to label the protein with isotopes in order to filter all the other contributions from the cells, and also to observe the signals with sufficient resolution in multidimensional NMR spectra. The way this is done is by growing human cells, HEK cells specifically, and then transfect the cells for every sample preparation. This is a transient transfection, which provides good expression levels and very high efficiency of transfection. During the protein expression phase, the proteins are expressed directly in human cells, and the medium is replaced with the isotope-labeled medium. Typically, a medium which contains all amino acids which are labeled with nitrogen 15. In this way, we can provide a fingerprint then of the protein backbone by looking at the amide signals in the spectrum. And with this approach, it's possible to measure, to investigate protein folding and maturation, and also how mutations, pathogenic mutations, can affect these processes of folding or cofactor binding and maturation, and so on and so forth. It's also possible and quite straightforward to observe conformational changes which come from as a consequence of changes in the redox state, for example, the formation of disulfide bonds or other chemical modifications. And naturally, it's possible to study interaction between the protein of interest and native partners, which can also be co-expressed within the cells in order to compensate for the overexpression of the first. And also, and I will focus on this later in the second part, interaction with active molecules, compounds which we can provide to the cells, so like drug candidates or something like this. So this is I think, very promising for applications in the drug development. I've mentioned the isotope labeling. So in the ideal case, you do uniform label labeling of all the nitrogen atoms in the molecule and be able to observe the signals. But sometimes we need to make the spectra simpler. We need to perform selective labeling of some type of amino acids. Now, in human cells, this is surprisingly straightforward to do, because the majority of amino acids in human cells are actually essential. The cells cannot make their own. So we can prepare custom-made media where only one amino acid is replaced, is isotope labeled. In this way, it's possible to focus directly on the residues that we want to observe, and it simplifies the analysis of complex in-cell NMR spectra. These are a few examples. So it has been shown several times also in bacteria that methionines, for example, can be labeled. In this case, the methyl group at the end of methionine residues is labeled. Or also cysteines can be labeled, and this was an example we showed. It was very easy to follow the formation of disulfide bonds when cysteines are selectively labeled. In other cases, it's very useful to label histidine residues, and in this case, we're not looking at the backbone amide signals, but at the side chain of histidines. And in this specific case, these side chains are very close to a metal. They bind a metal in the active site of a protein. And so this turns out to be a very good reporter of ligand binding in cells. Now, it's also possible to make more advanced labeling schemes with amino acids which are labeled on the side chain, deuterated on all proton position except for some methyl groups. This has very good relaxation properties for NMR analysis, and also provides very high sensitivity in the spectra which are recorded. However, it's not always easy to find, to buy or to synthesize these selectively labeled amino acids. This is very often due to the fact that one of the synthesis, the steps in the synthesis has to make a stereogenic center. So to have stereoselective synthesis on a isotope labeled compound can become costly. Now, a solution to this is exploiting human transaminases. So transaminases convert keto acids to L amino acids with complete stereoselectivity, so perfect stereoselectivity. They are usually they are active in human tissues as part of the degradation of amino acids, but they are reversible. So the reaction they catalyze is reversible. That allows us to make them work backwards, provided the keto acid precursor that we want to transform in amino acid, and they will just do the work for us. And so in this way, the medium is a custom medium where the amino acid is substituted with a precursor with the desired labeling scheme. And this is a recent work where we demonstrated that this approach works and gives a very high quality NMR spectra. These are all spectra recorded on cells, on human cells, okay? They are overexpressing a target protein, carbonic anhydrase 2, as an example of the kind of labeling that we can achieve. So this is what is obtained by labeling the tyrosine, the keto acid that will become tyrosine, phenylalanine, which are two aromatic amino acids, and also aliphatic residues, leucine and valine, can be very efficiently labeled with these schemes. And in the spectra, you see a black, the overlay with the black one, which is the contribution from background signals from the cell, because they will incorporate these synthesized amino acids in other components. But then the colored spectra correspond to cells expressing the target protein. And as you can see, the signals are visible with very high spectral quality. Then we are pushing forward and try to expand even further these kind of special labeling schemes in human cells by, again, in collaboration with Robert Konrad and Roman Lichtenegger from the University of Vienna, who are providing us with the precursors labeled selectively on the backbone. The specific reason why we want to do this is whenever we want to investigate intrinsically disordered proteins, the spectral overlap becomes even worse than with globular proteins, because the chemical shifts of all different residues will fall in very, very close within, in a very, very narrow region of the spectrum. So we really need these specific labeling schemes. And it's possible to focus then on the backbone of the protein overexpressed in cells by using precursors, so alpha keto acids labeled on the position which will become the carbon alpha in the final amino acids. And then by incubating this medium, this time the medium is uniformly labeled with nitrogen 15, and then it's incubated together with the precursor on top of cells, which are just there to have transaminases do the work for us. And they will create this leucine, which is nitrogen 15 labeled and then labeled on the carbon alpha. And in this way, we can make spectra like this. This is a uniform nitrogen 15 labeled protein, but there are alpha carbons on leucine positions. So the normal HSQC will give us all the peaks from all residues, whereas specially filtered spectrum, which is actually a plane of an HNCA, where we filter based on the presence of carbon 13 on the alpha position, we can get much cleaner spectra where only leucine is visible and also the residue following leucine. So this gives also a good compromise between how many signals we see and how well dispersed they are in the spectrum. And I think this has a bright future for intrinsically disordered protein studies in cells and in principle also in purified form, because then we could in principle purify these. An additional type of labeling which could be promising, and we have been shown recently, is the introduction of fluorinated amino acids. This has been shown in human cells. So this has been shown for several aromatic amino acids, and it works reasonably well. Now, bear with me, these spectra were recorded on an old instrument, but now we have the newly installed QCI cryoprobe, which gives us much better sensitivity. Okay, so just imagine how less noise we can get nowadays. These were old spectra. And all these are different proteins expressed in human cells with two different amino acids, fluorinated amino acids. And you can see peaks, and they are quite well dispersed. They, even more interestingly, some proteins here do not provide a usable nitrogen 15 proton nitrogen spectra because they tumble too slow in the cells due to interactions. And with this type of labeling, we can see signals from proteins which would otherwise be invisible by proton nitrogen NMR. Furthermore, it's been shown that introducing a carbon 13 in the position which is covalently attached to the fluorine atom in an aromatic system can allow exploiting the Trosi effect between carbon and fluorine, further increasing the resolution and the sensitivity of spectra and allowing 2D spectra. And we show that this, with the same approach I just showed, is possible to achieve in human cells. So this is this is actually a lysate, but to show that the incorporation of a fluorinated carbon 13 labeled amino acid can give us this nice 2D spectra where we see signals from specific positions in the protein from human cells. Now for the second part, the application to drug screening. So as I said, I think this approach can be useful to provide information in a physiological context about the interaction between the target protein and a drug candidate or a series of compounds which we want to screen. So it gives us a possibility to perform a screening in cellular settings in the first phases of the drug development pipeline, so in preclinical phases. This is the target model we have used very often up to now, which is carbonic anhydrase. It's part of a family of 15 isoforms in human, and many of them are involved in specific different diseases. So it's also an interesting target. This can be labeled with nitrogen 15 and provide information from the moving from the shift of specific signals on the target engagement. Therefore, we can treat the cells with a compound and be able to tell whether this compound is bound to the target protein inside or not. It can also be done by looking at proton signals of the target protein itself. So it can provide a way to tell if these molecules that we screen can actually reach the target by permeating the plasma membrane and can actually bind the target. Sometimes it's not easy to observe directly the target. This is the case, for example, with the N-terminal domain of HSP90, which is a promising target for anti-cancer drugs. That's the protein. Once it's expressed inside the cells, it does not give rise to good proton-nitrogen spectra. This is the red spectrum. That's the in-cell spectrum. This is due to interactions with other components of the cell, leading to line broadening. In this case, we can switch to ligand-observed NMR, and specifically focused on ligands which contain fluorine atoms. Why is that? Because fluorine is very sensitive and it's not present in the cells. So we will have spectra which are clean from any signals from the cells. We will only see the ligand. And this makes possible to screen fluorinated drugs also on targets which cannot be visible by detected easily by NMR in cells, and can give information on the drug permeability, on the target engagement, and also on the binding to other components of the cells of the drug, off-target binding. So that's an example of what you get, a simple 1D fluorine spectra of cells expressing a target protein, HSP90, treated with this compound. You see one signal which corresponds to the complex with the target. For another compound, we get the same complex with the target protein, but we also get an additional signal, which is present also in a negative control, meaning that this drug is binding somewhere else, for example, to membranes. And this is useful information to get on a screening in vitro. And then in an even more recent application, we focused on different forms of carbonic anhydrase. They are all cytosolic isoforms, but some of them are important in different diseases. And it's important to develop drugs which are as selective as possible towards specific isoforms. As you can tell, the binding sites of all these isoforms are very much conserved, so they share the same structural properties. So it's challenging to develop selective compounds for these kind of isoforms because they are very similar to one another. We have developed a series of fluorinated sulfonamides, which all bind very efficiently with high affinity all the isoforms that we tested, which are cytosolic CAs, 1, 2, 7, and 13. In vitro, so on pure proteins, they bind with nanomolar affinity. And then we set out to check whether they would engage the target in cells. And indeed, most of them, all of them actually targeted the target once every single target was specifically expressed in the cells, and then the cells were treated with each of these compounds. So every spectrum you see is coming from a complex between one specific isoform and one of these drugs. It's interesting to see that the chemical shifts are almost always different from one another. That means that we can exploit the very good sensitivity, the very high sensitivity of fluorine chemical shift to different chemical environments to distinguish between different complexes, different ligands, different isoforms. What's more, we can also, we did design one of these with 9 equivalent fluorine atoms that gives us even higher sensitivity. So that means that we can really reach almost physiological concentration of protein if we want with this kind of synthetic compounds. And with this, we can then exploit the competition between 2 ligands on the same cells expressing the target protein to find out which ligand is stronger than the other. And in this way, we can actually make a ranking of the compounds based on the competition between pairs of compounds. So by treating 2 compounds at a time and then analyzing the cells, we can find out which compound binds with higher affinity within the same cellular settings. And in this way, it's possible to perform a qualitative ranking. In this way, we don't quantify anything, but we can tell that this compound is binds stronger than this. And this is okay. This is the complete set of data with all pairs of our compounds, of our fluorinated compounds on 3 different isoforms, for which we can tell that there is some isoform selectivity. Some of these compounds they have a higher affinity for an isoform compared to another. Even more than that, we can be quantitative. So we can make a quantitative analysis of the affinity of these compounds. For this, we have to switch to time-resolved NMR in living cells. This is done with an NMR bioreactor, which is implemented and available as part of the service of Incel NMR in Instruct-ERIC in Florence. It's a customized Bruker flow unit in which we put cells in the form of agarose gel containing the cells. Here is the cross-section of the agarose gel. And a flow of medium is provided with these channels in a way in order to provide fresh nutrients and make the cells, keep the cells viable for up to about 3 days of experiment. So that makes possible to measure in real time what happens when we treat these cells with a ligand, or actually with the 2 ligands. And this is how we do the competition binding. So in this case, we have a 2-channel pump, 2 reservoirs. In both reservoirs, there is a SPI ligand, which is our reference compound. And then in one of the 2 we also put the test ligand. And in this way, we can change the flows of the 2 reservoirs and control the concentration of the second ligand over time. This is the sample preparation. We put the cells in this agarose gel, and then we assemble all the bioreactor, and this is the scheme of a bioreactor run. So over time, the concentration of test ligand is increased in a stepwise manner, whereas the SPI ligand is kept constant. And then we obtain, assemble the whole unit. This is just a scheme of the 2-channel pump, which controls the flows, and that's how we decide how much ligand to put in the sample. And these are the cells in which the target is overexpressed. And that's an example of the kind of time-resolved NMR spectra that we can obtain. So this is over the course of about 70 hours. We see the increase in the bound ligand, which is so to the right is the SPI ligand, and to the left is the test ligand, which is displacing over time the SPI ligand. We can actually resolve the free compound, which is outside of the cell, and the bound compound for both drugs. In this way, we can quantify everything. This is the plot of the free compounds, and on the bound free SPI, bound SPI, which is displaced by the bound test compound. So it's then straightforward to obtain the ratio of the 2 dissociation constants within cells. And in principle, if one knows the dissociation constant of one of these compounds, can actually be quantitatively measure the dissociation constant of all the other compounds. With this, I will finish, and this is the current group of Incel NMR team in Florence. Azzurra and Martina just defended their PhD thesis, but they were instrumental in the work that I show you today. And these are new PhD students, right, working with us right now. And of course, I thank Lucia Banci and Letizia Barbieri, part of the team. So thank you all for your attention.
Grazie mille, Enrico. Very, very interesting and showing many, many possibilities and bright future for Incel NMR, actually. We have some questions. Maybe I see, John, I see that someone cannot access the Q&A queue. So can you check? In case, can you write the questions in the chat? Francesca, Karen, if you are interested in questions, you can write them in the chat. Okay, we have in the meantime a question from Stefano Morasso he wrote, thanks Enrico for the interesting and clear presentation. Is there a way to monitor and discriminate multiple binding events of a ligand with 2 or more targets by ligand observed in cell NMR?
Oh, so thank you for the question. So if I understand, the question is whether we can observe the binding to 2 or more targets simultaneously. And in principle, yes, if the chemical shift of the fluorine atom on the ligand changes enough when it binds to 2 different targets. And this is similar to what I showed with the carbonic anhydrase isoforms. So it really depends. So for some ligands, there was an overlap of the chemical shift, but in rare cases, in most other cases, the chemical shifts were slightly different when the same molecule would bind to 2 different isoforms. So I cannot tell for sure because it will depend on the chemical environment around the fluorine group. But if this separation is sufficient, then yes, we can tell if it's bind to target 1 or target 2. And concerning treating with multiple ligands, possibly this is even more powerful because different moieties with fluorine atoms have wildly different chemical shifts, like they change by tens of ppm. So in principle, we could treat the cells with a mixture of compounds, not just 2, but many, and see which ones bind stronger to the target. I hope I answered the question.
Okay, thank you, Enrico. And then we have another question from Francesca Pauletti. Thank you very much for the very insightful talk. Which type of information can you collect within cell NMR on secreted proteins? Maybe their maturation before secretion?
That's a tough question. I've never focused on proteins which are natively secreted outside of the cells. It's possible that we might get some information of the protein before it gets secreted. I would be worried about the homogeneity of the cell sample because at any given point in time, I expect to have some protein which is in a step of the maturation and one protein which is in another step between the endoplasmic reticulum and the Golgi, for example. So it would be then challenging to find out which specific points of the maturation we are observing. I cannot say no, so it will be great to try, of course, but I don't have direct experience with these kind of questions.
Okay, the very last question from Doriano Lambda. Great talk. What about allosteric ligands?
I guess the question is if it's possible to observe binding to allosteric binding sites. So again, in principle, yes. Then how weak these ligands can be for us to be able to observe the bound, the complex with the target, whatever the binding site is. So whether it's the active site of just a secondary binding site, that's another story. So very weak interactions might be difficult to observe, at least with the setup, with the approach that I showed. If this interaction is strong enough, it doesn't matter if the binding site is the active site or if it's an allosteric site. If it's strong, NMR will see it in this setup.
Okay, thank you very much again. And then let's go ahead because time is running. Thank you, Enrico. Then now it is the turn of Marco Fragai, who will tell about NMR spectroscopy to characterize antibody pharmaceutical formulation, expression of viral proteins for new drug development.
Wait for a moment. Try to how do you say, to
share your screen. Share.
Okay. Are you able to see the?
Yes, we do.
Okay, so
very well, thank you.
Okay, so good morning to everybody. I will describe briefly some services that are available here for the user at our center Instruct, and in particular focusing on drug discovery. So today, the biological therapeutics is the fastest growing family of drugs. And this family comprises several different classes of drugs, including monoclonal antibody drug conjugates, enzymes, other proteins, but also vaccines, nucleic acid, and viral therapy and gene therapy. But most of the biologics that are available for the clinical use are monoclonal antibody, about 70%. So therefore, the pharmaceutical company have developed a suitable analytical workflow for the determination of the critical quality attributes, in particular to determine the amino acid composition, the high order structure, the biological activity, the post-translational modification, and other critical quality attributes like, for example, the sterility, osmolarity, pH. Particularly relevant for us is the assessment of the preservation of the native high order structure. The IORD structure comprises the secondary, the tertiary, and quaternary structure, and is particularly relevant because we know that there is a tight relationship between the structure and the function of the proteins. And obviously the preservation of the native IORD structure is important not only for the pharmacological activity, but also for safety. The pharmaceutical company usually use several different analytical techniques to assess the preservation of the IORD structure. Some of them are low resolution techniques, like, for example, circular dichroism, Fourier transform, infrared spectroscopy, chromatography, and usually there is also in high resolution biophysical techniques, that is mass spectrometry. However, we know that there are other biophysical methodology for the characterization of the structure of the protein. Obviously NMR, but also cryo-EM and also X-ray spectroscopy. However, the characterization of monoclonal antibody with X-ray, cryo-EM, and NMR is not easy. The main reason, for example, for X-ray, the crystallization of the complex between monoclonal antibody and the target is difficult. And also the flexibility prevent the use of cryo-EM. Also for NMR, the characterization of this system is not easy. Why? First of all, because the molecular weight of the monoclonal antibody, most of them belong to the family of the IgG, and the molecular weight is around 150 kD. And obviously another reason is that the monoclonal antibody in pharmaceutical formulation are not isotopically enriched. Here at our center, we have developed a platform for the characterization of monoclonal antibody that use two different strategies. One that rely on the direct observation of the monoclonal antibody using NMR spectra in solution. The other that rely on the observation of the fingerprint left by the monoclonal antibody on the target protein. Concerning the first strategy, it is possible to record 1D proton spectra using suitable pulse sequence in order to remove the excipients that are usually present in the pharmaceutical formulation. That, however, these excipients are usually small molecules. So the signal can be, removed from the NMR spectra using specific pulse sequences. And so it is possible to collect 1D proton spectra on the different samples that should be investigating. And these spectra are compared with the spectra of one reference sample. Then the differences are analyzed using statistical methods. In this way, it is possible to, to monitor the preservation of the IORD structure of the investigated pharmaceutical formulation. Another more, detailed strategy rely on the collection of the hydrogen carbon-13 also fast HMQC spectra by observing the methyl region, exploiting the dynamics of the methyl moiety. In this way, it is possible to obtain a fingerprint of the IORD structure of the protein, in this case of the monoclonal antibody. Then the different spectra of the investigated pharmaceutical formulation are compared again among them and with a reference spectra, and using again analytical statistical methods, like for example the CCD, the combined chemical shift differences, or PCA, it is possible to to monitor the preservation of the native structure of the monoclonal antibody. In this respect, is particularly important the use of high-field magnet. Here we have an example of the also fast experiment collected at 900 and 1.2 gigahertz on the same sample. We can see that at 1.2 gigahertz we have a much better resolution, and this is important to to have a deep characterization of the of the monoclonal antibody in the pharmaceutical formulation. In this respect, we have an example in collaboration with the Merck CERM. We have investigated, we have used NMR in solution for to monitor force degradation, to perform force degradation studies. One monoclonal antibody, abituzumab, has been treated with hydrogen peroxide at different temperatures to simulate the long-term storage. And we have obviously collected 1D proton spectra and bidimensional hydrogen carbon-13 correlation spectra. Then the data have been analyzed using the different statistical methods. And obviously, we have seen that the NMR is a powerful tool for monitoring the preservation of the structure, but moreover, thanks to the availability of the assignment of the methyl region of two domain of the monoclonal antibody, it was also possible to identify the methionine that undergoes through oxidation, and also to have a quantitative information about the oxidation state of the different methionine. The other strategy, I told you before, rely on the observation of the fingerprint left by the monoclonal antibody on the target protein. So obviously, the monoclonal antibody cannot be expressed as isotopic enriched protein, but many of the pharmaceutical target for monoclonal antibody can be expressed in E. coli, isotopically enriched in carbon-13, nitrogen-15, and also with deuterium. In this way, it is possible to analyze the complex between the monoclonal antibody directly from a pharmaceutical formulation with the target protein. And so this information are able to provide the chemical shift perturbation. We have, in this respect, we have an example with, again, in collaboration with Merck CERM. We have investigated the interaction of a fusion protein that has been obtained by covalently link a monoclonal antibody with that as a variable part of avelumab. So this monoclonal antibody is able to bind the PD-L1, that is an important pharmacological target, with other two identical domains of another protein that are able to bind the second target on the cancer cell. So for this, for the variable part of the avelumab, in complex with the PD-L1, there is a X-ray structure that has been used as a reference. So we have performed carbon-13 detected experiment and proton detected experiment on the complex between PD-L1 that obviously was isotopically enriched with the fusion protein, and so we were able to obtain the chemical shift mapping and to identify the residues that are involved in the binding with the monoclonal antibody. And these residues match perfectly with that of the X-ray structure. And we have another application of this approach. For example, we have investigated the different binding mode of the three different monoclonal antibodies that are approved for the clinical use, again with PD-L1, and we were able to, we performed the chemical shift mapping for each of the monoclonal antibody interacting with PD-L1. We collected the proton detected spectra, and so from the chemical shift mapping, we were able to see that the three different monoclonal antibodies bind the PD-L1 in a different region of the protein. And this data match with the X-ray structure obtained by, by the complex between the variable part of each monoclonal antibody with PD-L1. The difference is that we are able to investigate the complex obtained by using the pharmaceutical formulation and not the protein expressed fragment of monoclonal antibody expressed for the structural studies. Finally, in the frame of the Isidore project, we have started to develop a portfolio of for the screening of ligand and drug candidates against flaviviruses and COVID-19. We know that flaviviruses and COVID-19 are a potential threat for the human health. And concerning flaviviruses, we have decided to select as a target the proteases NS3 that is responsible for the proteolysis of the viral polypeptides. This enzyme releases the single proteins that are needed for the virus assembly and for the virus release. And however, this protein is, how to say, cannot be folded by itself, but to form the catalytically active complex should bind the NS2B. Therefore, we have used two different strategies in order to produce the catalytically active protein. One that rely on the expression of a fusion protein where the NS2B and NS3 are covalently linked by a polypeptide linker, and another strategy that rely on the co-expression of the two domain NS2B and NS3 Pro. Concerning the Zika virus, we have already expressed the protein, we have already performed the experiment, and the assignment of the NS2B, NS3 proteases for Zika is already available and is available for the screening of by ligand-based experiment and protein-based experiment for the user. We have now working at the expression of the dengue NS2B, NS3 proteases, dengue 2 and dengue 1. The dengue 2 protein is already available, and we have already performed the experiment for the assignment of the resonances. And now we are already started the expression of dengue 1 that will be available soon. And we have just received the plasmid for the expression of the NS2B, NS3 for the West Nile virus. Concerning the SARS-CoV-2, we have 5 years ago, we already expressed and characterized the wild-type proteases, and now we have expressed and characterized two mutants, single point mutants of the protein that are pathologically relevant. All this last research activity has been carried out in the frame of the Isidore project, and all the proteins are available for the user that want to perform ligand screening or protein-based screening for the identification of new drug candidates. And thank you.
Grazie Marco, thank you very much. Questions from the audience? I don't see questions in the Q&A queue. Any curiosity concerning these applications? Okay, if not, then we can reserve some time later on, and we can pass the stage to Paola Turano, who will present a metabolism view of disease course, vaccination, and pharmaceutical treatment. Prego, Paola.
Yes, good morning, everybody. So I will show you, if I can, I will show you a few examples of the activity that we have.
We cannot hear you well. It's very, very...
Sorry?
We cannot hear you.
Oh?
I mean, it's a very weak.
I will speak loudly.
Excellent. Thank you.
Also stay closer because the sound is like it comes from far. Yeah.
Okay.
Thank you.
Okay, so I will show you a few examples of the activity that we are conducting here at CERM by ourselves, but also as services to users that are focused on the aspect of metabolomics that deal with biomedical applications. So with respect to all the instruments that Lucia was describing before, the standard for metabolomics are the 600 megahertz instruments because they represent the best compromise between sensitivity, resolution, but also cost for running experiments that usually rely on hundreds of different samples in order to gain statistical significance. So metabolomics is the closest omics to the individual phenotype and measures all the small molecules, where small molecules are defined as molecules with a molecular mass lower than 2000 Dalton, that are present in a biological matrix. This means that at variance with the other omics, we have a large variety in terms of chemical composition of the different molecules. And here is an example of the distribution of the different type of chemical compounds that can be measured in the human metabolome. The NMR accessible part of the metabolome is just a small subset of the entire metabolome because we are able to measure by NMR only those metabolites that are present in concentration above 510 micromolar. Still, this means several tens of molecules in a single sample. Which type of samples can we measure? We can measure cells, we can measure tissues, we can measure biofluids. There are differences at the information level that we can derive from this type of samples. In terms of cells, we can define the specific pathways that are altered in tumor cells with respect to healthy cells, so that can be altered upon treatment with drugs. When we move to tissues, then the situation is more complex in terms of interpretation, but usually tissues are reporting on the health status of a given organ or apparatus. The same holds for some specific biofluids that are confined within a certain apparatus like cerebrospinal fluid. Then the most standard samples that usually are used in metabolomics are blood, urine, or saliva, and this type of biofluids are providing information on the overall health status of individuals, but at this level is more difficult to reconduct the observed alterations at the level of the metabolome to a specific biological or biochemical pathway. So let's start with cells. Here I'm showing 600 megahertz spectra of 3 different cell lines, human medulloblastoma, murine glioma, and human ovarian cancer cells. Here you see what we can measure in the lysates. These are about 30 metabolites that are in common in all the cells because the main biological pathways are always the same. But you can see that depending on the cell type, we have very different relative concentration of these molecules. Equally important is what we can measure in the growth media, because cells are continuously exchanging metabolites, uptaking some from the media and releasing others outside from the cell. So the complete picture of what is going on in a cell during its growth and during treatment requires the measure of both the endo and the exometabolome. How can we use metabolomics of cells to evaluate, for example, the effects of drugs. Here is an example of what we have done on a small library of gold compounds that were used to treat ovarian cancer cells under equal toxicity conditions. And then by analyzing the changes induced in these cells by the different compounds, we can cluster them in different groups. Some that were inducing negligible effects, and a few others that are here characterized by different colors that were inducing significant changes, but with unique signatures. So there was a group that was mainly inducing changes in the levels of glutathione and altering a few other pathways. Another was mainly effective on the glycolysis, another was mainly affecting on the TCA cycle. So it is possible with this type of approach to screen libraries of compounds and try to classify them according to the main biological pathways that can be affected upon treatment. Of course, not all drugs are really significant at the level of metabolome. For example, if one analyzes the effects of very well established drugs like platinum compounds, cisplatin and its analogs, the effects induced at the level of metabolome are more weaker because the main target is DNA, so the main effects are not at the level of the metabolism, but more on the replication of cells and so on. While when you have drugs that are targeting specific proteins, then the effects can be very, very easy to measure. Then, as I told you, we can use also tissues. When we use tissues, we use intact tissues, which means that we use a few milligrams of cryopreserved tissues with an approach which is called high-resolution magic angle spinning NMR. So it's magic angle spinning as in solid-state NMR, as Lucia was describing before, but the sample are spun at very low rate in order to preserve their integrity, because these are tissues are semi-solid samples, and the type of pulse sequences that are used to record the spectra are solution-like NMR pulse sequences. The quality of the spectra is shown here for colon, heart, and liver. You see that again we can measure easily a few tens of metabolites. We can assign and quantify them. And so this can also be used to compare the healthy versus diseased tissues, ability to revert pathophysiological states that upon drug treatments in animal models, for example. So it's very easy and does not require any extraction of metabolites. So we have a 600 megahertz equipped with a CIRMMP probe, and this is very useful as a tool, especially when you have animal samples. Of course, great attention should be paid to the collection procedures, and so pre-analytical procedures, because when we collect samples, they should be treated in a way to reduce as much as possible alteration in the metabolome, so that what we are going to measure at the end represent as close as possible the in vivo metabolome. So this means that the samples should be collected and frozen quickly and with the right procedures. Then biofluids. This is a comparison between some representative samples. We have CSF, the bottom panel, and you see there are several metabolites. A few of them have been labeled, but only a few. Again, we are about 30 metabolites that can be easily assigned and quantified. We have urine, which is an easily to be collected sample, but as an intrinsic intra-individual variability, day-to-day variability. So usually to extract the individual fingerprint or the fingerprint of a disease, it is important to collect from the same individual multiple samples on different days. Then we have serum or plasma, blood serum or blood plasma. These are the most common biofluids that are used in metabolomics because they can be collected easily. It's minimally invasive collection, and they are more stable than urine, so a single collection from an individual is relevant, and this provides a stable fingerprint of the individual metabotype. As you can see, at variance with the other two biofluids, in serum we have several broad resonances that overlap with the narrow signals from the small molecule metabolites. These broad resonances are due to the presence of protein and especially of lipoproteins. So they are also informative on the status of the individual. And we have a 600 megahertz, which is standardized and equipped with everything which is necessary to use the BI Lisa Bruker tool, which is a tool that allows to quantify not only metabolites but also these lipoprotein components, which are about more than 100 parameters that can be measured in a single sample. So this is very informative because having in a single spectrum both the small molecule metabolites and lipoproteins provide a very well-defined picture of the healthy status of an individual. And so just a schematic picture, if I have an individual and I make multiple collection of his blood on different days over time scale that could be also of years of time scale, you see that I have an intra-individual variability that is smaller than the inter-individual variability. So each individual has its own specific metabotype space, which is different from that of the other individuals. But why metabolomics can be important to study diseases or to study treatment? Because upon disease onset, the metabolome of each individual undergoes a shift in the same direction. And so it is possible with statistical tools to extract which are the meaningful changes at the level of metabolome which are characteristic of a given disease. On the other hand, if I follow treatment, I can monitor the effect of treatment on each individual, where the best treatment is that is able to bring back the new phenotype, which is characteristic of the disease, to the original healthy phenotype. A nice example on how this was possible is represented by COVID-19. There was a big effort at the level of the scientific community to understand whether metabolomics could be used to learn something about this disease already from the very first wave. And here I presenting some results of a project that was financed by Regione Toscana, where we analyzed more than 500 individuals that were collected, whose samples were collected at the moment of hospitalization, and then we had information on the course of their disease, whether they develop a mild or severe conditions, in some cases a fatal outcome, and then some of them were also resampled after recovering, a few months after recovering. And you can see that we have a very good discrimination, more than 93%, between the COVID-19 subjects during the acute phase of the infection and the recovered subjects. But even more, if you look at this box plot, you see the gray band represents the normality values from a large set of pre-COVID healthy individuals. And you can see now in this number of metabolites like mannose, phenylalanine, and glucose in this example, we have a slight drift in the values for the those individual who had a mild infection, more significant changes for those who had a severe disease, and those that were going to have a fatal outcome were those with the mainly dysregulated levels of these metabolites. During recovery, this is the yellow box, the individual revert back towards the normality band. The effects are also present for a number of lipoprotein components, which are represented in the box plots at the bottom of this picture. So we could use this type of analysis also to follow what happens during the healing process. So people were sampled at the moment of the hospitalization, then more than 21 days after the infection, and then again post-COVID, a few months post-COVID. And what we could see is that those individuals that have been collected after 21 days from infection are represented by gray dots, and at the level of metabolites, the values are reverting back to those of the recovered subjects, so they go back to the normal values, while at that time point, the lipoproteins that are dysregulated are still at the values of the acute infection cases. So this means that the lipoproteins are slower in reverting back to the normal values. This type of approach was also used to follow the possible efficacy of treatment with tocilizumab during the first wave of COVID on a small set of subjects when people were trying to identify drugs that could control the infection. Then, always during COVID, we analyzed the effect of the Pfizer-BioNTech vaccine. You know that there was a lot of discussion about possible side effects of the vaccine. So we received a few samples from our colleagues in the local hospital. They had analyzed over a time scale of almost 2 months, the response of individuals that were treated with this vaccine. And we had 10 individuals that were COVID naive, so they never get in touch before with the infection, and other 10 individuals who had recovered from COVID when they got the vaccination. And as you can see, these green bands represent the dysregulation induced by vaccine during this timeline. At the level of the lipoproteins, because none of our metabolites that are accessible to us was dysregulated by vaccination, but there was clear dysregulation at the level of lipoproteins. That was very significant for those that were COVID-19 naive, but was less meaningful for people that had already been in touch with the COVID. The biological significance of this observation is not clear, is not obvious, but this is again represents a nice example of how we can use metabolomics to follow not just disease onset or medical or pharmaceutical treatment, but also development of protection. And this is the case that the example that I'm going to provide you for what we did in Isidore. But first of all, I would like to show you a comparison where we have different type of animals, human, non-human primate, and sheep as an example of a farm animal. And we wanted to point out that there are similarities, but of course also difference in the type of profiles that we acquire for serum samples of these animals. But for all of them, we have small molecule metabolites, but also all these broad components that are labeled in the top one that are derived from the different signals of lipoproteins. The system that has been developed by Brucher for the quantification of the different lipoprotein components has been developed and validated only for humans at the moment, but there is interest also in developing something for animals, because even in animals, the lipoprotein represents an important part of the blood profiles. What we have done in Isidore, we received a few samples from of macaque, so 4 animals that were treated with a malaria vaccine but maintained under chloroquine protection. Other 4 that were stimulated with the vaccine but were then developing also infection at the level of blood, and then control animals. And again, what happens in our analysis is that we found differences at the level of the lipoproteins in these animals in the first phase of the treatment, and then with time these differences are reduced, while in the control animals nothing significantly happened. Clearly, this is a small example because 4 animals for each group is very small for a omics approach, but still there is something that could be promising. And this is just an example to say that animals are also something that we can analyze with our approaches and instrumentation. And with this, I finish my example. I thank all of you for your attention, and I thank all the people working in metabolomics at CIRMMP.
Thank you very much, Paola. We have, oh, what is this noise? I don't know, we are almost running out of time, so I don't know if there is time for a quick question or 2, or if we should just say goodbye, because we had 1 hour and a half. John is appearing. Okay, if not, if there are no questions, let me just thank you all the participants and to all the speakers for this very nice, interesting webinar on a wide range of applications of NMR spectroscopy, and also additional biophysical techniques that can be integrated to NMR spectroscopy to provide really nice pictures of biological problems. And John, if you share the slide, let me conclude by advertising the upcoming webinar that is dedicated to the themes of fairy tale. And it will happen just in a few days from now, on December 16 at 11, as usual. Please join us with this very interesting webinar conducted by Antonio Rosato and Andrea Valsesia. And with this, I thank you all for your attention.